And today I want to tell you about glycans and glycobiology, and specifically data science and machine learning and how that can be applied to glycobiology. And I have a few introductory slides about glycobiology per se, because it may not be on the top of everyone's mind, and I want everyone to be on the same page for that. So we start very, very broadly. What are glycans? Glycans are a biological sequence, a biopolymer, and they are in some ways connected to the other biological sequences, to DNA, RNA, and protein, but not in the traditional sense that these other sequences are connected with each other. So DNA flows to RNA, flows to protein, which is the central dogma of molecular biology, of course. Glycans, in theory, flow from proteins, but not in a template-based manner. So glycans are built up from cascades of enzymes that take and remove or add monosaccharides to a growing glycan chain. If you want to put it really simply, glycans are basically carbohydrates or sugars. So long chains of sugars that can branch off in different directions and that can adorn proteins, and they can be on top of lipids. They can occur by themselves if you talk about bacteria, if you talk about plant cell walls, etc. So glycans are a universal biopolymer. To my knowledge, we haven't uncovered a form of life that doesn't have any glycans. So in that sense, it's as universal as the other biological sequences. And what is interesting is that it's a lot more information rich. So even though the sequences themselves are typically a lot shorter than you would expect from DNA or proteins, the diversity within this sequence can be a lot larger. And you see that here with the log scale chemical information content. And I'll go a bit more into detail why that is the case. So what I always find funny is that basically everyone works with glycans, but a lot of people don't realize that they do, because they are so ubiquitous. So I mean, if you want to be really technical, both chitin and cellulose are glycans in a sense. So by mass, glycans make up the predominant amount of biomass on our planet. Here you see a bacterium, a textbook depiction of a bacterium. This is how a bacterium actually looks like, or at least one bacterium looks like under an electron micrograph. These fuzzy hairs that you see are basically a decent part glycans. So every cell that we know of, so this is not only true for bacteria, this is true for basically every kind of cell, is covered in this sugary forest that also has as a consequence that every interaction between these cells and every interaction that molecules have with cells have to involve or at least be aware of glycans to some extent. If you want to drill down further into the more molecular area, we can look at proteins. This is the spike protein of coronavirus, the protein that it actually uses to make contact with our cells and subsequently invade our cells. So this would be a typical textbook depiction of a graphically rendered protein. This is how the protein actually looks like. It is an averaging of a molecular dynamics simulation of the glycans in dark blue that are covalently attached to this protein. So you see that across a certain time window, most of the surface of the protein at some point is covered by a glycan. And I mean, this is actually, in a way, it is an underestimate, because you can see that the glycans are only depicted as a ball and stick representation, whereas the protein has a surface covering. So if you would also depict the glycans as surface covering, this would be even a bit more covering. This is really important because you see there are certain spots that are never covered by glycans, and these are of course very important for designing antibodies and recognizing antigenic epitopes of this viral protein. And this, by the way, is not only true of the coronavirus spike protein. This is true of most proteins, especially for the proteins that are secreted or are exhibited on membranes. So for humans, for instance, well above 2/3 of all human proteins are likely to be glycosylated, and this number actually increases all the time because people uncover new forms of glycosylation. What I find funny is that even this picture, even the picture on the right, is an underestimation of the glyco world, because the lipid membrane that you see on the bottom, of course, also would be glycosylated because you have a lot of glycolipids, etc. So this is even a more impressive picture if you would include all forms of glycans. So for me, when I entered glycobiology, it was a bit like the movie They Live from the 80s, if you're old enough and know that. The protagonist there finds a pair of glasses that when you put them on, you see how the world actually is. So you see the subtext behind ads, etc. So for me, it's like I looked at biology a certain way, and then I uncovered the Are Glycans, and now I look at biology a different way. Glycans are not only a lot in terms of both abundance and diversity, they also basically exercise a lot of functions in biology. And I just picked at random 2 very important functions, but there are, of course, an enormous number more. So one would be the interaction with our microbiome. So all our mucosal surfaces predominantly consist of glycans in a sense. So mucus itself is basically mucins, which are proteins that display glycans. So most of the mass of mucins are made up by glycans. And these long sugar chains interact with our microbiome, and that can have a range of different interaction modalities. So they can be a source of nutrition for certain bacteria. They can also form points of adhesion for certain bacteria. So in a way, you select, you shape the microbiome, and you prevent malicious actors from getting a hold of your gut, for instance, but also other mucosal surfaces. Recent papers also have shown that there are more molecular very relevant functions, in the sense that some glycans, for instance, down-regulate virulence functions of parts of our microbiome via interfering with the quorum sensing process and other players here. Another part, I mentioned that the cellular surface of basically all cells is covered by glycans, and that means that basically all pathogens that have to interact with cells typically also interact with glycans, and that includes basically all viruses. That includes bacteria that adhere via adhesins, etc. But it also includes toxins and also the normal cell-cell communication pathways as well. And I'll talk a bit more about viral cell interaction via glycans later on. I just wanted to show you basically to depict this again with the coronavirus, because it's very current, of course. So everyone probably knows by now that the coronavirus interacts with ACE2 as a protein receptor, but what people tend to forget is that every cell has this glycocalyx, this sugary forest of glycan trees, and there is no real way, if you draw it to scale, that this virus can come to the cell without having to touch these glycans. So and indeed, there are known interactions that have to happen prior to binding to ACE2. So in the case of SARS-CoV-2, it is definitely established that it binds to heparan sulfate, which is one of these extracellular glycans, and there are even reports that it binds to other glycan moieties as well before transitioning onwards to ACE2. So just if you go even more toward the atomic scale, what are glycans on a chemical level? And they are, as I mentioned, they are these chains of sugars, of simple sugars that are called monosaccharides, and you can string these together via different means. So you have a vast library of monosaccharides. An interesting property of glycans, by the way, is that they have a non-universal alphabet. So we all more or less have the same 20 amino acids. So humans, mice, bacteria, etc., with very, very few deviations. With glycans, this is decidedly not the case. So the pool of monosaccharides is very much species and taxonomic group dependent. And what's more, you have different ways of connecting these monosaccharides. So you have basically between 12 and 20 different ways of connecting them, depending on which hydroxyl group you choose to connect the next monosaccharide to, and if it's in the alpha or beta configuration, basically, as you can see here. And that leads to a different structure in the end, a different 3D structure here, if you want, that has different properties. And this overall leads to this insane diversity. So what we want to do basically with our research is to take this diversity and find some meaning in this diversity, and ideally in terms of functions or properties that are associated with that. And I want to show you this brief vignette here of is that even a feasible thing to do? And this is from the immune system, and basically in a lot of different facets of the immune system, glycans play a huge role that starts from cancer, that uses glycans to evade the immune system by recruiting immunosuppressive proteins that bind to certain epitopes, but it also modulates, glycans also modulate the activity of antibodies and whole immune system cells. And so therefore they are really important for properly tuning this whole machine. But so this is known, and the part on the right side is also known. This is a very specific case. This is the family of glycans that can be present on immunoglobulin G, so IgG, which has a very conserved glycosylation site and on its constant part. But of course, it depends. So there can be different glycans can be attached to this glycosylation site of IgG, and that in turn affects the properties and functions of IgG. And in this paper here, they just annotated a few of these structure-function relationships that have been teased apart throughout the years. So the most striking is the loss of Foucault, so this red triangle here that leads to a drastic increase in antibody-dependent cytotoxicity, which is really important for all these recombinant antibodies that are used commercially for therapeutic reasons. So you can, by glycoengineering, you can to a certain extent tune the function of your antibody. But what is important is that this teasing apart of these functions is a huge amount of work. This is a collation of probably years of dedicated work of finding this out, and it's not really transferable. So if I give you a new protein, you can't tell me what specific glycan forms will do to its function. And if I give you new glycans for the same protein, you probably also won't be able to tell me what it does. And this is where we want to come in. This is a gorgeous illustration made for a Quantum Magazine article that was written about our work recently. And what we want to do is we want to basically read this language of glycans and see what effects, what functions does it entail, and can we modify that in a predictive manner to modify the properties and functions of biomolecules. We started to do that with a language model, actually. Language models by now have a tradition in biology. They have been used both from a natural language processing perspective as well as from a deep learning perspective for DNA, for RNA, for proteins with great effects. By now there are humongous models out there that work with billions of protein sequences, etc. And we want to do the same for glycans, which also are a biological sequence. They are nonlinear, if you haven't noticed before, which is a tricky part. It also increases the diversity, but it makes it a bit tricky for left to right reading language models, if you wish. So that's why we chose to sort of extract structural context from a glycan sequence to use that to read from left to right to predict a glycan property or glycan function. For the connoisseurs among you, it is basically an LSTM-based recurrent neural network that we use for this. One application that you can do with that is predicting glycan immunogenicity to humans. In this case, glycans are a immunogen to a certain extent, both IgM predominantly, but also there are more and more IgG cases that are popping up, so IgG molecules that are specific for certain glycan epitopes. A lot of allergens are glycan-based, and if you want to be really technical, even blood groups are immunogenic because there will always be a certain fraction of the population that has antibodies. And I myself tend to forget this by now, but many people don't know that the blood groups themselves are glycans. So you can see here human blood group B, which is a specific glycan, and the same holds true for all the other blood groups. So we basically we took a set of glycans that has been ascertained to be that people have antibodies against that, and trained a model to predict based on sequence is a glycan immunogenic or not to humans. And the representation that the model learned is on the right. So every dot is a glycan, and they're colored according to the label. And you can see, the first thing that you can see is that the model clearly can separate these two categories quite well. And even among the categories, different classes of glycans that share, of course, sequence similarity cluster together. And what was important for us here is that O-linked glycans and glycolipids are somewhat overlapping with the immunogenic glycans, because those are the glycans that are in direct contact with bacteria, and bacteria typically mimic these glycans to sort of fool our immune system, and therefore you have this overlapping area. We we did this language model with quite some success. We like what we got, but the problem here is that glycans are not a linear sequence, right? So we ran into troubles in the sense that we had to find workarounds of how to deal with that, because you have these branches. So we did this compromise of extracting structural context. But if you actually think about it more if you put it on its head, basically, what a glycan looks like is a tree, right? And trees are nothing else than a certain class of graphs, where every node would be, for instance, a monosaccharide, and then you connect them via different edges. And luckily, there is a lot of progress on graphs, so we could piggyback on graph-based neural network that have been developed for actual small molecule drugs, so for chemicals, where either every atom is a node, and these kind of approaches have been shown to work really well compared to traditional handcrafted features, the chemical descriptor-based models. Or you can do the approach on the right, where you take specific chemical groups, so let's say rings, aromatic rings, or certain functional groups, and add that as a vocabulary, and that will be closely associated with glycans, because then in this cluster graph that is shown, every dot here would correspond to a monosaccharide, for instance, that would of course correspond to a chemical structure, but we would have then this nice graph. And we did that basically. We did graph convolutional neural networks with our glycans, which are of course a more natural fit to these branched sequences. And basically we did two things with that. So apart from showing that it indeed performs a lot better than the language models on the existing tasks, we wanted to sort of show that we can separate different taxonomic groups, because they have just different alphabets, because they have different glycan sequences, based on these learned representation from our neural network. And we also wanted to predict viral receptors, because I mentioned to you that viruses use these glycans as a kind of entry port into cells. So we wanted to see if we can predict from protein sequence which glycan epitope it will bind. So for the first part here, this is a model that was trained to sort of differentiate taxonomic groups, and the representation that it learned, you can clearly see that these via kingdoms it separates quite well. And even if you go below that, so some of the what seemed like disrupted clusters are actually phyla, for instance, that are inside one kingdom, and that are separated as well in the representation. So there you can learn quite a rich representation from this from these graph sequences, basically. And then sort of like as a last application here, with predicting the viral receptors, what we use as a data source for that is glycan arrays. So this is a technique where you couple hundreds of glycans to glass slides, you add a protein to this glass slides, or a whole virus if you want, that is labeled, and then it's essentially like a microarray, in the sense that you then look for glowing dots on this plate, and you know which glycans are where, so you can reconstruct basically the binding behavior, and use that to find out binding motifs for certain glycan binding proteins, such as viruses. So we basically had this model. It's sort of like a matching model. So basically it takes a virus or a viral protein. In our case, we work mainly with influenza virus, so hemagglutinin would be the relevant binding protein here, and a glycan with our graph convolutional neural network, and we ask the model, does this lead to binding or not? And so this is how we trained the model, basically, based on these around 120,000 measured protein glycan binding interactions. And this was really nice. So we did this mainly for influenza virus, because we had most data there, of course. And we could recover basically all the literature known binding motifs, and predicted a few new ones that are quite similar in terms of sequence, at least. And we also could extend this to other viruses. So for instance, here, rotavirus, which is a common infant targeting virus, and we did this for several others as well. What we want to do now, basically, and this is sort of like a briefish outlook before I close this, is to sort of both standardize this and make this more approachable, because this is it is quite a new field in this sense, and we want people to be able to use these techniques very easily, without having to develop them from scratch by their own, and without having to be software or without having to be data scientists, etc. So we want to, ideally, we would want bench scientists to work with these techniques. So we did a Python package called Glycowork, where we have most of these functions sort of in a more user-friendly way. And the whole idea of Glycowork is that users, if they wish at least, they can work with this human readable format. So here it's depicted as a graph, but what Glycowork uses are these string-based glycan names, which are typically not nice to work with computationally because they are ambiguous, non-deterministic, etc. But internally, we convert everything to graphs, and then once you have that, you can basically piggyback on the whole graph literature, which is amazing. I mean, this is a huge field, and then you can do all kinds of really cool things. For instance, detecting motifs via subgraph isomorphisms, and we do that in this position-specific manner, so that we, for O glycans, for instance, that we only detect the core motifs if they're actually at the reducing end, and not if they're somewhere in the sequence. And this is quite efficient, at least for the amounts of data that glycobiology typically offers. And with that, we can, for instance, automate these glycan array analysis. So to get significant binding motifs of this of these viral proteins, etc. We can also enable sequence context analyses. So here would be one of these embeddings that is learned by one of the deep learning models. Everything is stored as static files in Glycowork, so you can just get this type of embedding for a set of sequences that you input, and then analyze how a sequence context of a certain monosaccharide in a specific group of organisms, etc. looks like. Here, for instance, rhamnose is a typical monosaccharide that occurs in quite homogeneous sequence contexts. So you see that, for instance, D-rhamnose typically is connected to other D-rhamnoses, and rhamnose is connected to other rhamnoses. So that's why you have this sort of homogeneous cluster in the embedding space. So everything I showed you up to now is basically was done with this data set here. So we work with about 13,000 species-specific glycans, 19,000 unique glycans. What makes me very optimistic about the future of the space is that what we are currently working with internally are these numbers. So 23,000 species-specific glycans, 47,000 unique glycans, and we are in the process of generating even the next data set that is even bigger, and some of the differences are shown here basically. So this is with the growing amounts of data, of course, we can do more and more things that are really cool, a lot data hungry models, and I think this is quite the exciting space for the years to come. And with that, I already come to my acknowledgement. So I would like to thank the team at Harvard, of course, for working on the Glycan language model. The graph convolutional neural networks were done in collaboration with John Quackenbush's group at the Harvard Public School of Health. And then, of course, I would also like to thank my own group who started at the beginning of this month and who will probably push this a lot farther than I did. And I would like to thank you for your attention.